Good morning. So today our topic is all about bacterial cellular morphology and simple stains. So for the background, of course, we are familiar with wavelengths, and there are different wavelengths uh, in visible light. And for example, we have here a leaf, and of course, a leaf contains a lot of pigment called chlorophyll. The chlorophyll could absorb red and blue light, and the effect is some of the wavelength are to reflected back to our eyes. And those wavelength that are reflected back is kinda the color of that pigment. So we perceive the reflected light as a color. So for example, in this color wheel or color pie chart, uh, if a if a substance absorbs mostly red color, so the or red wavelength, so usually we perceive that substance as green. Okay. So whereas if it's absorbed majority like blue wavelength, so we perceive that substance as or like reddish orange. Okay. So usually we use pigment for staining, and for microbiology, we all know that under the microscope, if you want to view bacteria and some microorganism, we need to do simple staining, at least simple staining. Why? Because under the microscope, they are too thin and they appear like colorless, so it's very difficult to see bacteria and other microbes under the microscope without stain. Okay, so this is an example of staining procedure, and this is called Van uh, K. Gyson. I don't know the correct pronunciation, but take note this procedure they use three types of stains. Here they have hematoxylin, which stains the nucleus and it appears bluish. The dotted structure here, or the dark structure, those are actually bluish when we try to magnify this tissue. And of course, we have acid fuchsin. This one stains the cytoplasm, muscle, and erythrocytes. And of course, we have picric acid, which could stain collagen in this tissue. So take note, um, we tried to combine different kind of stain to make a complex staining procedure, and of course to increase differentiation, like to differentiate one cell to the other cell. So again, as I told you, that viewing cells with standard light microscope is so difficult. That's why it's essential to use stain uh, for contrast. So once stained, we could identify the cell morphology, size, and of course the arrangement. And usually stains are solutions. Yeah, they are made up of solutions. So therefore, there is a solvent. And usually we use water or ethanol as a solvent for our pigment or colored molecule. And most of the time, those colored molecule is made up of aromatic carbon, or we call it benzene derivative. Later, I'll show you. Okay. And those colored molecule, we call it chromogen. Okay. So again, so we define stain as an organic compound, usually contain benzene ring plus a chromophore and oxochrome group. So what is a chromophore and what is a oxochrome group? So again, we have here, of course, basic, which is benzene. This is an organic colorless solvent. And if we add chromophore to our benzene structure, um, that's the time um, we have a colored compound. Okay, so technically, chromophore plus the benzene structure, that would be equal to chromogen. So chromogen is the colored compound, not a stain. Okay, and then we add the oxochrome. So what is oxochrome? So oxochrome, it's a chemical group that convey the property of ionization to the chromogen. So enabling it to form salts and bind it to fibers or tissue. So therefore, our oxochrome, these are kind of charged molecule. It could be negative or positive. And therefore, the oxochrome also gives the properties of your dye, whether your dye is a basic or acidic. Okay. So there are some important chromophores. So these are important chromophores. So if you see C double band C, C double band and C double band O, C double N double band and NO2 and quinoid ring. Those are example of um, chromophores. Okay, so here we have two benzene ring, and then this benzene ring is connected by uh, nitrogen. Like there's two double bond between the nitrogen. Therefore, this nitrogen group is our chromophore. And then there is another nitrogen here, like with excess, nitrogen with two lone pair electron. I think this nitrogen is important to form a bond. So another characteristic of oxochrome, um, it also intensifies or increases the ability of chromophore to accept um, or absorb energy. Okay, so therefore it's responsible for darkening of the color. Okay, so for to make it clear, for example, we have here benzene, um, and then if we add three nitro groups or the chromophore, we will have trinitrobenzene, which is actually yellow in terms of color. Okay, and if we add another oxochrome hydroxide, we will form a trinitrohydroxybenzene, okay, which is yellow stain. We call it picric acid. Like I showed you before, we use sometimes picric acid in histological um, studies. Especially, we use picric acid to stain collagen uh, in the tissues. Okay. Okay. Another example, we have methylene blue. And as you can see, the backbone of methylene blue usually absorb around 440 nanometer. Okay, but if we add some functional group, we kind of increase its ability to absorb more um, energy. Okay, so now we have 585 nanometer. It means it intensifies or strengthen, or usually it darkens the color of the stain. And if we add more functional group of nitrogen, we further increase the energy absorbing capacity or of uh, our dye. Okay. So example, again, this is methylene blue, and we could use methylene blue as a simple stain. Okay, so when we say simple stain, we only use one stain, or um, we only use a single reagent. So to produce a distinctive contrast between organism and background. So we just wanna classify uh, here uh, bacteria versus the background. Okay, of course, using simple stain, we could get some information about our sample. So for example, the cell shape, the morphology, yeah, the cell shape or the morphology of the cell, as well as the arrangement. So in this example, we have uh, a rod. So this is actually rod or like bacilli, but it's hard because it's only 400x. And if we further magnify, this is according to them kind of rod structure, but still it's kind of difficult. But as you can see, it's kind of longer, so that's why it's rod in shape. Okay, and I don't see any arrangement of bacteria here, so I think these are just solitary uh, bacillus. Okay, so okay, another thing, this is 1000x, remember? And this is quite big, and these are actually not bacteria, but of course yeast. It also uses methylene blue to stain the yeast. So methylene blue is a basic dye. Okay, so if it's basic, it usually um, it's a positive charge, and it usually stain the negative part of the cell. So if there are some proteins or some part of the cell that are negatively charged, the stain usually stay there. Okay. So DNA and RNA, they are negatively charged in nature. That's why if we put methylene blue, the methylene blue will goes to DNA and RNA. Okay, so usually it goes near the nucleus, since there's no nucleus for bacteria, so it stays to the cytoplasm of bacteria because the DNA is in the cytoplasm. Okay, and yeah, here yeast, I think it goes to the nucleus of the yeast, and as you can see, it seems like it's cocci, but the difference is that um, yeast usually under microscope they're kind of oblong in shape, oval in shape, and technically bigger than bacteria. Okay, and this saliva stained with methylene blue, of course, uh, these are like squamous cell of uh, buccal cavity or yung cheek cells. Um, so malaki yung cell, cheek cell, and here, as I told you, methylene blue usually goes in the nucleus because there's high presence of negative charge molecule, which is your DNA. So this is the nucleus. And the cytoplasm here, yung light blue. And as you can see, we have here some, like not some, a lot of bacteria. Okay, so these are actually bacteria stained by methylene blue. Okay, another stain we have here, safranin. As you can see, the charge is positive, so therefore safranin is a basic stain. So it also goes to the nucleus or stick with the RNA or DNA or any negative um, components of the cell. And here, itong picture na to. So do you think these are bacteria? No, actually it's too big for bacteria. So as you can see, this is nucleus. And I think this is a dividing cell. So these are actually the chromosome. Okay, so I just wanna uh, show that safranin has high affinity with um, the genetic material or the DNA. Okay, so these are the chromosome of dividing cells. So it looked like bacteria, but no, it's too big. Okay, and then we have another stain here. Okay, here we have positive charge oxochrome, and therefore if it's positive charge it's basic, and malachite green, and we use malachite green to stain endospores. Okay, so later I'll explain why we use malachite green to stain endospore. Of course the color is green. Okay, then we have crystal violet. So again, here we have positive charge, and therefore crystal violet is a basic stain. Okay. What about eosine? Do you think eosine is a positive or negative stain? Okay, so since eosine is conjugated with sodium, and we all know that sodium is um, like positive, so therefore this oxygen is negative, like they make this like a salt, like, so therefore eosine is a negative stain. Okay, so rose bengal. So there is sodium here. We all know that sodium is positive, so it will form complex with negatively charged oxygen here. This negatively charged oxygen makes this pigment acidic. Okay, and of course from it named acid fuchsin, so therefore it's acidic. Why acidic? We have here negative um, charged oxygen, and we can make salt by, of course, calcium ions. Okay, what about carbolfuchsin? So here we have positive functional group, positively charged functional group, and we are actually making salt, like uh, we have chloride here. So therefore, this positive charge functional group makes this uh, carbonphosphine basic. Okay, so therefore this is a basic dye or basic stain. And carbonphosphine, it's important uh, for acid fast staining. Okay, sometimes we cannot differentiate bacteria if they are gram positive or gram negative, like they could be gram positive and gram negative at the same time. So it's not effective for some bacteria to classify them into gram positive or gram negative. So we use acid fast staining or Gill Nielsen staining. I don't know if I pronounce it right. Okay. So later I will explain what is acid fast staining. And so that's the end of this presentation. So just remember, we use simple staining to differentiate the background as well as the cell. We just want to increase the contrast to determine the arrangement, morphology of the cells. Okay. And another thing, most of the examples are positive staining. Why positive staining? Because this stain or dye, it goes inside the cell, and we're kind of visualizing the cell itself. If we wanna, if negative staining, so negative staining, usually it stains the background and the cell is kind of colorless. So that is what we call negative staining. So later I will explain what is the purpose or function of negative stain.